we have to talk about is where we get our fats. The diet is where we get the majority of our fats, and this is going to be through ingestion, right? So we're going to eat something yummy and delicious with high in fat, and what's going to happen is that that fat is going to cross the intestinal mucosa. And then what's going to happen is it's going to be packaged, most commonly with proteins. We're going to talk about what those packages are here in a moment, and that's going to allow us to move our lipids through the bloodstream, and then it's going to be delivered. And this is where I would normally say, where could it be delivered? And hopefully you guys come up with two places. One, the adipose tissue. This should make sense because this is the storage place for our lipids. Or it could go to the liver. And a couple of you guys have asked me about fatty liver before. And the reason our livers are so important is because it's for the oxidation or repackaging of our fats. So really the liver is important for how we're going to break down our fats. Before we talk about breaking down our fats, which I'm going to do in class, what I wanted to do was wanted to give you a quick review of our lipids. Okay, so lipids, by definition, they all have one thing in common. They're all insoluble in water. So because they're insoluble in water, it makes it difficult to transport them throughout our bloodstream. So really, in order to transport our lipids, we usually have to require some sort of carrier protein that's usually attached to our lipids. And so an example of that would be our albumin is a carrier protein, and we're going to talk in detail about how this actually works. Vitamins, they can be carried by other things, like for instance, vitamin A is carried by a retinol binding protein. Steroids tend to be carried by things called steroid carrier proteins. And so in order for the lipids to be transported, they almost always are attached to some sort of protein. So again, up here, if we look at this particular one, here's a bunch of lipids, right? How are they being transported? By something called apoprotein B, right? And so this is just an example. So the bulk of our body's lipids is cholesterol, is phospholipids, like what's in our membranes, and triglycerides. And so these are transported in the plasma by large complexes called lipoproteins. And so this is an example of a lipoprotein. So it's how we're going to transport, and you can see all three of them. So our plasma membrane is going to surround the ones that are really hydrophobic, like triglycerides. They're going to be in this nonpolar core here. So in our lipoproteins, there's a structure to them. I kind of already hinted to that. So the first thing is that the structure is that it has this core, that's the inside here, and that's hydrophobic lipids, and then they're going to be surrounded by our phospholipids and our cholesterol, right? So our glycerol, would be attached on these, and then our proteins, of course, are on the outside too, right? So the reason is our hydrophobic, we're going to hide it away from the water. So these proteins often contain cell targeting signals. So they're going to have, again, those proteins on the outside, something that helps to get them where they need to go. And these lipoproteins can have different densities. So the higher the protein content, the higher the density of the lipoprotein. So lipoproteins are classified based on their density. So the first example is something called a chylomicron. So chylomicron is an example. So again, here is an example of a lipoprotein. Chylomicrons, as you can see here, these move lipids through the bloodstream, and really it's the very general term of packaging with a protein. So when lipids are packaged with a protein, they're called chylomicrons. Then what we have is we have things called VLDLs. So VLDLs, which are very low density lipoproteins. Then we have IDLs, which are intermediate density lipoproteins, and LDLs, which I think is one of the terms that you guys are probably familiar with, low density lipoproteins, and then HDLs are high density lipoproteins. So these are all examples. And again, for all of those lipoproteins, their structure is going to be, a hydrophobic core consisting of the triglycerols and cholesterol esters, and then that's going to be surrounded by this amphipathic phospholipids or unesterified cholesterol, things that are going to be able to interact better with the water on the outside. So what is the function of this. So the function of our lipoproteins is that cells can't actually synthesize, most cells can't synthesize their own cholesterol. So how are they going to get it? Well, we have these lipoproteins that are floating around in our blood, and so what we're going to be able to do is that a receptor protein for a particular cell is going to say, hey, I'm a little light on cholesterol today, right? So it puts out the receptor, one of these lipoproteins is going to bind to it, the two membranes are going to fuse with each other, right? And then cholesterol is going to be able to enter into the cell that needs it because we have our cholesterol esters on the inside here, right? So plasma membranes can fuse and then it can release that cholesterol. Pretty cool, right? So again, the issue with this, so we have this range that we're supposed to have. So cholesterol, we're supposed to have this range for females and males, it's slightly different. And then again with the HDL and LDL. But issues can occur when there are genetic disorders. So one of the ones is familial hypocholesterolemia. And what happens is the receptor protein doesn't work. So no receptor protein means we can't get the LDL inside. So what ends up happening is if we can't get these LDLs to release the cholesterol inside the cells, then there's going to be a really high level of cholesterol in the blood. And when we have a high cholesterol level in the blood, that's going to lead to atherosclerosis. So with this genetic disorder, there are actually 5 classes of mutations that have been identified with this disease. So in some individuals, they don't have the receptor at all. So it's just not synthesized. So if you don't have the receptor, you're not going to be able to bind to the LDL, you're not going to be able to pull in the cholesterol. Sometimes they have the receptor, but it's not transported to the surface of the cell. Because remember, our receptors are proteins, right? And so proteins are made inside the cell and then they have to be transported. Some of them have the receptor and it made it to the surface of the cell, but it just fails to bind LDL. Some of them have the receptor and it binds to LDL, but then what ends up happening is it fails to actually pull the cholesterol into something called a clathrin-coated pit. So this is how we actually bring this inside. And then, of course, some of them go through everything. So they have the receptor, it binds the LDL, it goes through this clathrin-coated pits, but yet it fails to release the LDL inside the cell in the endosomes, which is how we bring things in. So that's an example of one genetic disorder with this. The other one that we often hear about is HDL, which is our high-density lipoproteins. So sometimes cells can take up more cholesterol than they need, right? So too much cholesterol in our cell would be bad because then we would have too fluid of a membrane. We would have, remember this is the basis for a lot of our hormones. So to get rid of the excess, they give it to the HDL. So LDL drops off cholesterol, HDL takes away excess cholesterol. So what we're going to do is we have transporter proteins in the membrane that's going to move the cholesterol from the cytosol to outside where the HDL picks it up. So commonly HDL is known as the good cholesterol. And it's not actually good cholesterol, right? Cholesterol is cholesterol. But what it is it's actually the transporter of the plasma cholesterol back to the liver. So the function is to transfer these cholesterol, to pick up these lipids, and to either take them back to the liver or transport them to other lipoproteins. But the overall reaction is it gets it out of the bloodstream, it gets it out of the cell. So that's why we like to have high levels of this HDL. So, of course, there's a genetic disorder associated with this one also, so this is Tanner disease. And what happens is that this transporter protein doesn't work. So we end up getting cholesterol buildup in the cells. So I think you can imagine at this point that that's going to disrupt the membrane structure. So again, this is the typical, and I should say for ages 15 to 29, I tried to pull the ones that are most relevant to you guys. So what are some of the ways that we can avoid high LDLs or low HDLs, right? So one of the best ways to do that is just regular, vigorous exercise. So when you regularly exercise, you're going to raise your HDLs. That's the good cholesterol, right? And then and not only if you exercise, but if you have a low-fat diet, avoiding red meats, you're going to reduce your serum cholesterol levels. So that was just my brief review as we went through. Again, an lipoprotein, which is what we have here. This is just a lipoprotein that also contains some sort of apoprotein in the outer shell. And the apoproteins have important roles in lipid transport and in metabolism. So again, they're going to have a structural domain, because they're proteins that are going to be recognized by those cell receptors. Usually they're alpha helices that's going to interact. So as we go through, again, this is most commonly how lipids are transported in the blood through that chylomicron structure. Again, the idea is that we need to have the correct levels of our lipids, not only in our cells, so we don't disrupt cell membrane, and in our blood, because too much can lead to, the hardening of our vessels and arteries, right? So that could cause heart attacks, but also in our liver, because that's where we're actually going to then package it to be stored. So this is a really important system. But the point of talking about all of this is to get you to this. Why do we care about fatty acids, and especially breaking them down? And that's because oxidation of our fatty acids is the major energy yielding pathway in many organisms. So this is where I'm going to pick up in class. We're going to go talking about why there's advantage to storing energy as fatty acids, and then ultimately we're going to talk about how we break them down. So I will see you in class.